episode 183 of the Stem Cell podcast, Glomerulus-on-a-Chip, with Dr. Samira Musah. Hey everybody, we are Daylon and Arun. Welcome back to the Stem Cell podcast, where we culture knowledge in stem cell research by talking to some of the brightest minds in the field. A quick note about our upcoming schedule. This is our last episode before the holidays, and we will be taking a short break. But don't you worry, because we'll be back on Tuesday, January 12th, with a brand new episode. So make sure to mark your calendar and check back in then. Today, we have Dr. Samira Musah from Duke University, Go Blue Devils, on the podcast to talk about her research, bioengineering functional models to study human kidney disease. We've also got our usual roundup of recent highlights in stem cell news that's coming right up.
But first, and yes, go Blue Devils, looking to stay current with the latest research and news in the field of stem cell biology, we'd like to remind our listeners to check out Stem Cell Science News, featuring the most recent top peer-reviewed research and review papers, as well as industry, policy, and science news. Stem Cell Science News provides a platform that allows researchers to stay up to date with their field while saving time. So subscribe for free at stemcellsciencenews.com.
Arun, I'm kicking it off today with the lymphatics. The lymphatics are very hot. No one was paying much attention to them back in the day. It was all about the arteries and the veins, but we've heard enough about those two cousins. Now we're talking about the lymph almost every day. And, you know, owing to that attention, the molecular functional characterization of lymphatic vasculature has significantly improved. And we know a lot about it. In the heart specifically, we know that formation of new lymphatics, or lymphangiogenesis, delays our atherosclerotic plaque formation. It facilitates the healing process after myocardial infarction. And this suggests that, stimulation of lymphangiogenesis, making these new lymphatics in the infarcted heart, could improve cardiac function. And this is supported by studies in mouse and zebrafish that show that the newly formed lymphatics, they provide a route for clearing immune cells in the injured heart. And that's part of what promotes cardiac repair. But whether lymphatics have additional functional roles during heart development and cardiac repair is not known. You know, the idea is that it's just like a path, a conduit. But, you know, angiocrine, as Shaheen would say, there may be some functional or instructional input there that's driving the process. So this is a story from Guillermo Oliver, who's at Northwestern. Also on this paper is Paul Burridge, your boy, Arun. You know, they were interested in identifying what are the molecules coming out of this lymphatic vasculature, the lymphangiocrine signal produced by these lymphatic endothelial cells. What they found that there was, in fact, a secreted factor that was specific to lymphatic endothelial cells that controlled proliferation, also survival of cardiomyocytes, and that's during embryonic development. But also they showed that the lymphatics were important during neonatal cardiac regeneration. You know, in mouse, you can cut off a big chunk, like 20% of the heart, and it'll regrow with no scar. So lymphatics are important for that process. Also, the lymphatics are cardioprotective after myocardial infarction in adults as well. That's what they show. In development, when they get rid of the lymphatic endothelial cells, the hearts are small. And that's because there's reduced cardiomyocyte proliferation and increased apoptosis, and that's important. We're going to come back around to that. And then by taking primary cardiomyocytes from heart and putting them in conditioned media from lymphatic endothelial cells, they showed that it was a secreted factor that was mediating this proliferation and increased survival. And then they took a mass spec approach to characterize the secretome from the conditioned media of those lymphatic endothelial cells, and they identified reelin as a specific protein that underlied the process, or was involved in the process at least. They knock out reelin specifically in the LECs and show that they recapitulate that small heart phenotype, that you need reelin for efficient heart repair after the neonatal infarct or injury. And here's the big kicker. In adults, they used a collagen patch that was imbued with reelin and showed that it could improve heart function after myocardial infarction in adults. So there was, I think, what brought it to the caliber of nature. It was a real therapeutic idea there that you could use Reelin in a patch and give it to patients. So yeah, a big story in the heart. Arun, I'm surprised you didn't jump on this. I got to it first, I guess, but I think it's an important story. Anytime you can identify a specific factor. I love angiography, you know me, but also a therapeutic factor that could be delivered in a practical way. I think could be a huge step towards meeting the needs of a huge class of patients. You know, cardiovascular disease is the biggest, right?
It is the biggest, but I am trying to expand my horizons, Daylon. You know, if you haven't noticed, I haven't covered a cardiac paper too recently.
I have noticed.
Or have I? Yeah, see, I'm trying to expand my horizons. I'm trying to be a good trainee and learn about things outside of my field too. So, yes, but hey, I'm still a big fan of the work. It's a powerful study because it tells us a little bit more about the lymphatic system, and it seems like this is a really hot topic. We've actually covered a couple of similar-ish papers here on the podcast in the months past. If you remember, we covered Sherry Gur-Cohen's and Elaine Fuchs's paper that came out in Science, where they actually found that the lymphatic system has a pretty important role in skin hair follicle regeneration, and you know it's a cool model system for studying that. And, you know, Paul Burridge is on this paper, a former guest of ours. So there was an iPS cardiomyocyte role in here, and it's cool that they were able to identify this secreted factor in Reelin that actually has this positive impact. And like I said, you know, it's a really cool dual role for the cardiac lymphatic system. It's got this developmental role in regulating developmental formation of the heart, and also this, you know, injury system as well, this injury model. And so it's really the lymphatic, you know, system is more than just a drainage system. It's important for homeostasis of cardiac and, you know, other organs as well. So there's a lot we need to learn about how the lymphatic system actually works throughout the body.
Yes, for sure. Shaheen, this is a nod to you. Angiography. But I will say there's a couple things I have to say about this that there's open questions. One, you mentioned the Fuchs skin story. Well, you might be interested to hear that the source of cells, so they got commercially available dermal lymphatic endothelial cells. That's what they used in the co-culture, you know, to generate that conditioned media and then to identify using the mass spec from the whole secretome. That was from conditioned media from dermal lymphatic. So I think while Reelin clearly is able to have an effect there and is a potent, perhaps therapeutic, I wonder if we've really captured the totality of the lymphangiocrine secretome. And I wonder if there aren't other factors that really are unique to the lymphatics in the heart. So I'm guessing that we're going to see another story soon enough that focuses on the lymphatics specifically in the heart. And maybe identify some extra factors there. But, you know, like you said, like I said, this is really a cool story, both from the developmental, you know, just basic insight, but also from a clinical standpoint as well. So very exciting work.
Good stuff coming out of Guillermo Oliver's lab over there in Northwestern. Side note, has your guy Shaheen Rafi been on the podcast?
He has been. We're going to get him on again. He had a story a little while ago that was pretty big with these ETV2 endothelial cells. But, you know, I think it's time. Shaheen, you got to come back. You're going to come back on for us, buddy.
I just want to hear him say that angiocrine in his very specific voice.
Are you saying my impersonation isn't adequate, Arun? Come on. I've been studying this guy for a decade.
I'm just saying I haven't heard the real thing, so I don't have anything to compare it to. How about that? All righty. So we're going to move from nature paper to nature paper. Next up is a story coming out of the Harvard Medical School. We're talking about the eye. So we shift from the heart to the eye. Reprogramming to recover youthful epigenetic information and restore vision. Pretty simple but very powerful approach coming out of David Sinclair's lab, right down the hallway from where I used to work, actually in Boston, in the new research building, I believe. So we know that aging is something that, unfortunately, all of us going through right now. I think all of us in 2020 are going through a form of accelerated aging, if you know what I mean. So we're talking about aging and, in particular, the retina as a model for aging. First author on this paper is Yuanchen Liu. So we can use the eye as a good model system to study aging and degeneration of the nervous system. And this paper is really cool because it's really the first successful attempt to reverse glaucoma-induced vision loss rather than just study its progression. Okay, and this is definitely going to be hopefully reproduced in other organs as well. But the general premise here was that the Sinclair lab used a cocktail of factors that all of us in IPS biology are very familiar with. That's OCT4, SOX2, and KLF4 to actually restore youthful methylation patterns and transcriptome in mouse retinal ganglion cells, both during aging and also after an injury phenotype. So what they did, it's a very straightforward but beautiful study. They used AAV2 to actually deliver the OSK, everything outside of MYC. The four canonical Yamanaka factors are OCT4, SOX2, KLF4, and c-MYC, right? But they didn't introduce c-MYC here, obviously, because it has some oncogenic potential. But they found that just 3 OSK were enough to actually promote axon regeneration and retinal ganglion cell survival after an acute optic nerve injury. Okay, so that's in itself is very powerful, that after an injury, you can actually use the OSK to restore a healthy phenotype. But then they kept on going, and they found that it's actually the DNA methylation that's required for this OSK-induced regeneration phenotype. And not only that, 4 weeks after expressing OSK, the vision loss was reversed. These mice actually started seeing again, and they started seeing well. So even after this quote-unquote glaucoma mimicking damage has already happened, 4 weeks after that's happened, you can still use OSK to restore vision in these mice. And finally, the restoration of this youthful vision is it's a methylation effect, right? They further confirmed that, and they also restored vision in old mice. Okay, so not only in this acute injury system, but this OSK was able to restore vision in older mice to a signal of youthful vision. So it's I think a very powerful study because it's showing the power, the effect of OSK, 3 factors that we all know about, in reversing not only aging, but also in injury phenotype in the eye. And the natural next step here is to ask, okay, if this works in the eye, what other tissues could OSK turn back the clock on, right? So I think there are a lot more studies that are soon to come out of the Sinclair lab, which is really world known for its focus on aging.
Yes, you can imagine the headline. I mean, you saw the headlines when this story came out, and I want you to try to imagine how many. I'm sure you got the same phone calls from all your, the older people in your life whose vision is failing, and they read the headline. They're like, "Hey, I can." They did it in mice. Obviously, no one's going to be injecting any OSK into their eyes, and I'm not trying to say that was like the end point here. I doubt that Sinclair would claim that is the therapeutic approach here. It's more understanding, as you said, that the methylation really is the ticket. I think the irony here, though, is that Sinclair, as you alluded to or stated explicitly, is famous for the aging, but like specifically the study of aging and the mitigating the pathology surrounding aging using resveratrol, which was the last thing that everyone was like, hey, I could just pop a pill. And I think the irony there is that was like a global, like, hey, you take this one factor and it pretty much turns back the clock on everything. And that was a while ago that was a big splash. And now he's been, I'm sure he's still going along that path too, but now we're going in kind of the other direction. We're talking about cellular reprogramming and like Yamanaka type stuff, which I wouldn't say is diametric opposite of resveratrol, but it's certainly in a different sphere. So Sinclair is clearly expanding his horizons like you, Arun. We're all trainees at the end, right? Forever.
Mm-hmm. Forever learning. But hey, I mean, I actually think that maybe there is a translational orient to this and a translational angle to this. Maybe this is something they really are pursuing. I mean, yes, of course, you have to be extremely careful with introducing OSK into anything, right? It's extremely powerful, an extremely powerful effect, as we're seeing with iPS reprogramming. But if the effect is this dramatic, then perhaps it's worth taking a closer look at when it comes to actually using it clinically. I don't know.
Maybe, but I'll say this on the air. Nana and Pop, I know you're not listening, but if you are, don't go to any stem cell clinics and get some OSK directed into your eye. We know how that ended. We talked about that in the past, about these rogue clinics that were injecting mesenchymal stem cells into the eye. Do you remember that? And this is a good segue, actually, because I got a story about mesenchymal stem cells. In that case, it was a bad outcome. And go listen to the episode and you'll be reminded. We're not going to talk about that. We're going to talk about something positive with MSCs. We've been positive on MSCs for a while now. People are looking more closely at these cells, whereas before they were pretty loosely described. Everything was an MSC and you take it from anywhere, inject it everywhere. But we're looking more closely, okay? We're getting a more granular view. And, it's important in the blood, in the marrow specifically, because while we've focused for decades on understanding the origin of all these, myeloproliferative disorders and, cancers in the blood, hematological malignancies, we understand the blood element of that. The niche is less well understood. Or we understand the niche really only as a driver of normal hematopoiesis, right? How does the niche support hematopoiesis? But there's another element there where in disease conditions you often get bone marrow fibrosis, okay, where the normal hematopoietic cells and niche cells are replaced with scar tissue, okay? Deposition of extracellular matrix, all the hallmarks of fibrosis that you see typically, oftentimes downstream of mesenchymal stem cells or myofibroblasts, et cetera. And what this is accompanied by is a loss of normal hematopoiesis, and then you get this extramedullary hematopoiesis as the spleen takes over and gets big, right? And as a downstream consequence of that fibrosis, the hematopoietic cells get all screwed up too, right? So we've understood a lot about those hematopoietic alterations, but the alterations in the non-hematopoietic compartment, we don't know that much about. And what we do know, though, is that there's like a morphological evolution. Looking at the bone marrow of these patients, you can see that there is like a pre-fibrotic phase, and it undergoes this stepwise progression to full-blown fibrosis. But, the initial changes, the molecular events that underlie that switch and progression, again, unknown, okay? Not characterized. So this is work coming in by Rebecca Schneider and her lab, who's at Erasmus Medical Center in the Netherlands. And what their approach was to try and understand what are the relative contributions of mesenchymal stem cells, because we know in solid organs, mesenchymal stem cells are often what drive fibrosis. So they went into the bone marrow and they said, what are the cells here? What are the cells they're doing? They've done previous work looking at these Gli-positive myofibroblasts, that here they went to look at these cells and say, are there other cells that are also contributing to this? And what is the molecular signature and mechanism underlying this pre to post fibrotic transformation? What they found was that there were two distinct MSC subsets that are pro-fibrotic, and that they kind of described their evolution. And the real key here, which I think is what made it into a cell stem cell paper, was that they found this kind of druggable component here. They found that the alarmin complex, which is made up of S100A8 and S100A9, that was a signature of progression in a murine model, And also, they found that there was a signature in patients when you look at their stroma, also in the plasma. So it's like a circulating marker. So that's a nice outcome here in terms of biomarker and diagnostics. And then they show that there's this small molecule tasquinimod that inhibits the alarmin signaling, and that was able to rescue the fibrosis in the murine model of this. So that was the big splash here, is a therapeutic. Identifying that this is the factor, this alarmin is a factor in the process, but also showing that it could be druggable and targeted, I think is what is going to make this a highly cited paper moving downstream and maybe entree to clinical trial.
Yeah, neat study. And like you were saying, I think the reason why this was in Cell Stem Cell is because not only did they have a really nice basic approach, but they had the rescue with the tasquinimod, right? So that's always a huge plus. We were lamenting before the show about how the numbers for some of their single cell analyses were pretty low, but again, I've said this before, I'm a big fan of the limitations section that the Cell Press journals have thrown into all papers now. And actually, the reason why their single cell numbers were pretty low is apparently liquid bone marrow is really tough to aspirate in advanced myelofibrosis. So they're not able to get too many cells for that reason. And so they're saying straight up, they're saying that, yeah, we know this is a problem. We know we only got a few hundred, maybe 1,000 cells for our analysis, but we're going to expand the data set in a later cohort.
Yeah. I mean, hey, I wish I could get my papers through with that. Well, a lot of we're gonnas. My editors don't listen to my we're gonnas, but I guess it works for Rebecca Schneider. I do agree, though, there's fundamental limitations when you're working with patient samples, right? So I think they're off the hook there. But even so, I mean, I'm not being actually critical here, but it's just an open question for me as someone who just, I had a story just now with a bunch of single cell, and it's not the numbers. I mean, not for nothing, but we had over 100,000 cells. But I'm not flexing on number of cells here. I think that's silly. But what I will say here is that when you have limited cells and in a mouse system where I think maybe that's controllable, I think you have to be careful with the Seurat. The Seurat does this unbiased clustering analysis. And that was kind of what identified the original populations of the story. They were like, we've got these 1,000 cells, which is not a ton, to be honest. And they were like, we did unsupervised clustering, and it showed us these 6 or whatever populations that we subdivided. So, I'll just say that when you do the Seurat, oftentimes, no joke, Arun, you do Seurat on your computer, and then you do it on someone else's computer, the same script, and the UMAP plots look different. I'm not saying that the clustering is different, but there's nuance here. And I don't know that I'd trust a computer program that's still I wouldn't say it's in beta, but it's constantly being revised to tell me the biology. But I will say it gives you an amazing lead. And the key thing there, though, is to validate and to corroborate and verify on the back end. So yes, I think they'll expand. We're gonna expand the patient cohort. I think we need we're gonna on validating those cells, isolating them by independently corroborated markers that show up in seq, pulling them out of there, and then doing some more functional analysis. And I'm sure that's going to be forthcoming from the Schneider lab.
All I got to say, Daylon, is don't forget to update your version of R, okay? When you have problems from your computer to my computer, a lot of times that's what's going on. It's a different R version, which having done and having dealt with Seurat in the past, that's a lot of times that is the issue, which is very painful because you got to go back and reinstall all your packages, and I don't want to think about that right now. Moving on, Nature Biotech paper is our last paper for the Roundup. This is coming out of another lab that's right down the hallway from David Sinclair's lab over in HMS Genetics, the one and only George Church lab. First author here is Alex Eng, who I believe is a graduate student in the Church lab. I think we overlapped for a little bit when I was in Boston. The title of the paper is A Comprehensive Library of Human Transcription Factors for Cell Fate Engineering. This is a really neat study because it's using human pluripotent stem cells to improve a whole slew of differentiation protocols, and in particular, transcription factor isoforms. And they were able to basically develop something called the human TFOme, the human transcription factor ohm. Not the catchiest name, but it's a comprehensive library that contains like 1,500 transcription factors and 1,700 transcription factor splice isoforms. And they screened the library across 3 different pluripotent stem cell lines and discovered that just around 290 transcription factors are able to induce differentiation in, get this, 4 days. As short as 4 days without actually altering soluble or biomechanical cues, like, for example, like throwing small molecules on there. So just by introducing select transcription factors into pluripotent stem cell culture, you can get a very fast, very effective, dramatic differentiation phenotype into a variety of different cell types that you're interested in. So they actually use 4 of their hits in their TFOme to reprogram pluripotent stem cells into neurons, fibroblasts, oligodendrocytes, and also something that you're interested in, vascular endothelial cells, or endothelial-like cells, that are actually molecularly and functionally similar, not only to what you would get from a normal differentiation protocol that would take weeks, but also pretty similar to primary cells. Okay, so it's a cell autonomous approach, and it actually, the other cool thing was it enabled parallel programming of pluripotent stem cells into multiple cell types simultaneously. So if you mix and match the transcription factors that you're actually throwing into your pluripotent stem cell culture, you can get very effective but very selective differentiation of just 3 or 4 different cell lineages in a single population. So really powerful. And they included some oligodendrocyte inducible pluripotent stem cells. They mixed actually these oligodendrocyte inducible pluripotent stem cells with unmodified PSCs to actually generate cerebral organoids, which they showed that could actually generate myelin as well, also very powerful. So it's a really powerful approach, and I love this because it's improving differentiation, something that we're all trying to do. A lot of us just mix and match different growth factors, small molecules to induce differentiation down a lineage of interest. But of course, you always have a mixed bag of cells, right? You never have 100% of the cell that you're really interested in. But they're showing here that it really just comes down to the genetics. If you can find a master regulator, a master transcriptional regulator, such as an ETV2, an NKS3-1 that they show here, then you can get the cell type that you want in the course of days, as opposed to weeks. And I'm sure you have a lot to talk about when it comes to this particular study, especially the ETV2 angle, because I think it follows up on a paper that you actually worked on in years past. Is that right?
Yeah, we had a story about direct reprogramming of amniotic cells to endothelium, and ETV2 was the primary inducer there. It's the master regulator. And Shaheen actually just had another story. We should have him on about that, with ETV2 making these kind of pan-endothelial cells that could do everything, super cells. So yes, not surprisingly, ETV2 was potent here in direct induction, but I think the nuance that was nice is they showed that it was this one isoform in particular that led to like 99% versus the other isoforms that were either, not effective, 2 of them, or one that was mildly effective. So that kind of actually was revelatory for me, because we had some complaints about people not being able to reproduce the level of reprogramming that we were able to achieve. And I think it may have been because they were using the wrong isoform, which we actually raised that possibility in the past, but we got a lot of grief about it. Talk to George Church, people. And speaking of George Church, I mean, this is something that only could come out of the Church Lab, in my view. And I love it, but I'm in 2 worlds. There's like that part of me that's like, you got to grow the thing. And this is the alternate of that. This is where we're going to make the thing, and we're going to make it so robustly and with such precision and specificity And we're going to do it on a scale that's insane. I mean, the fact that they could throw this panel, they talk about like, oh yeah, we just made this screen and came up with 300. I mean, who's like checking out each one of those things? Your boy, that poor grad student suffering. But, only in the church lab where they have the money and he has that mad scientist kind of ethos that led, remember they had the galloping horse that they programmed into E. coli. This is the kind of stuff that comes out of the church lab that no one would ever think of, but everybody, perks up when they hear the story because it's like out of science fiction.
Church lab at its finest, right? This is the same lab that's working on bringing back the woolly mammoth. And I don't know how that's going, but that's another one of their claims to fame. And hey, he might be a poor grad student, but perhaps not for long, because in typical church lab fashion, they did start a company based on this paper.
Of course. Probably his 20th or more company. Another connection, church lab, our guest, Samira, she was co-mentored by Dr. Church, so maybe she can give us some insight into that mad science. But before we get to that interview, I got a message from STEMCELL, who would like to introduce their one-stop resource for researchers who are using or looking to use organoids in their experiments. STEMCELL's Organoid Information Hub provides scientists with instructional videos, educational webinars, expert interviews, technical tips, and curated publications to help researchers set up and optimize organoids as a research model in their labs. Learn about organoid culture from the experts at STEMCELL. Visit www.stemcell.com/discover-organoids. All right, guys, today I am delighted to introduce to you Samira Musah, who is Assistant Professor in the Department of Biomedical Engineering, also Associate Professor in Medicine in the Division of Nephrology at Duke University, mine and Arun's alma mater. So this is a special episode. We're doing a Blue Devil thing here. The Musah Lab is interested in understanding how molecular signals and biophysical forces guide organ development and physiology, and how these processes can be therapeutically harnessed to treat human disease. They focus on engineering stem cell fate for applications in human kidney disease, extrarenal complications, and therapeutic development. Dr. Musah, thank you so much for joining us today.
Thank you so much, Daylon and Arun, for having me on this show. I'm very thrilled to be here, and knowing that this is all Blue Devils on the show right now, it's super exciting.
Yeah, thanks for joining us. And so you are a new PI at the one and only Duke University, right? But you've been focusing on pluripotent stem cells for a while now. So for more than a decade, actually starting off working on synthetic polymers and hydrogels to control stem cell proliferation and differentiation. But lately, your claim to fame has been your work focusing on the in vitro disease modeling side of things, right? And particularly working on a tissue type that doesn't get a lot of love on the show, and not a type that not a whole lot of stem cell biologists work on, which is the kidney, right? And so a lot of snooty biomedical engineers and biomedical researchers might view the kidney as little more than like a simple filtration system, but it's so much more than that, right? So as someone who is helping to carry the torch for the kidney disease modeling field, and before we actually get into your two Nature papers, could you tell us generally why it's so important to study diseases of the kidney?
Yeah, thank you for, you bring up really, really good points about the field of kidney and nephrology and understanding human kidney biology. So I actually think that one of the reasons why the field has not been studied as extensively as other areas in stem cell biology is also because we have very little understanding of the human kidney biology and how the organ develops and its function. So those challenges also make it a little bit perhaps sometimes less attractive to some scientists, or maybe a bit more challenging generally to approach how to differentiate stem cells into kidney cells, and just imagine trying to model human kidney function outside of the body, because unlike a lot of other tissue types, with the kidneys, you're talking about a highly vascularized tissue that also requires very specific fluidic circuit. So just having our standard traditional tissue culture environment might not accurately model a lot of the complex functions of the kidney, which are also very important for disease progression. So I think these complexities also influence the decision, or at least some of the interest in that field. On a more, from a practical standpoint, I think that understanding human kidney biology is important for so many diseases. We know that most patients with, or a significant number of patients with diabetes also have kidney disease. And a lot of the drugs that actually are meant to work on patients with heart disease actually work on their kidneys. Drugs don't work directly on the heart. So it's important to understand how the kidneys work and how they influence the function of other organs, so that we can better understand diseases of not just the kidney, but some of these other diseases that are so widespread. Some other important factors that most certainly influenced my decision to go into the field is the fact that kidney disease is so prevalent. I think most people, myself included, before I started working in this field, didn't even realize how prevalent kidney disease was, or still is. There are more patients suffering from kidney disease than diabetes, COVID-19 disease, HIV/AIDS, and other diseases all combined. Worldwide. So it's quite a prevalent disease. And I think that we have about more than 10% of the world population suffering from the disease. And in the United States, it's actually even worse, with more than 14% of the adult population suffering from kidney disease. And there are no targeted therapies. So it certainly is important to understand human kidney biology. I could also kind of just add a little bit about how it remains very challenging to understand this in a way that's relevant to humans. So we know that unlike a lot of other some other organ types have actually been much easier to study, relatively, of course, easier to study using animal models. The kidneys have been one of the organs in humans that remain very challenging to actually understand biologically how it develops and functions by using animal models, primarily because we already know that there are divergent pathways in how human kidneys develop than mice or other animals' kidneys develop and function. So most of the time, the pathways that are identified simply do not apply to humans. And so it's been very challenging to extrapolate the results from traditional ways of doing things to human kidney disease. And so it really is important. And I think the stem cell field is probably one of the best tools we have to help understand some of these processes in a way that's directly relevant to humans.
Wow, you make a very strong case. There's a lot of people that just, if they were leaning towards the kidney, now they're, full bore. But let's talk about disease. There's a lot of diseases that arise specifically from the kidney, and then as you mentioned there's all these conditions that kind of converge on kidney dysfunction and or failure, and very, massive prevalence, so all these probably would benefit from like a regenerative approach. And I know we talk about how you're focused on modeling, and we're going to talk about your Nature papers in a minute with the kidney on a chip. But first, what are like the obstacles to therapeutic application of iPS-derived kidney tissue? Looking at the literature, I see that the differentiation protocols seem pretty tight. We just had Matthias Lutoff on the show. He was talking about like the synergy of bioprinting and organoids, taking these two principles of, printing as well as self-organization and converging. We had Sergio Pasca talking to us about how he's combining multiple organoids to make these assembloids. So all these things are coming together. What are the major obstacles left for applying this therapeutically?
Yeah, I think in the kidney field, it's certainly not. We're really not at the point where we have full understanding or have models that fully recapitulate entire functions of the human kidney yet. So I think as attractive and exciting as things like kidney organoids are, they do have many of the limitations that just about any kidney organoid has, in the sense that, which includes the fact that they are usually mimicking very early stages of development, typically about the second trimester. So we know that a lot of kidney diseases are postnatal in nature, right? So it requires having models that can more closely mimic the mature and functional state of the kidney. So our lab is definitely making advances in that with specific focus in the glomerulus. In fact, we developed a method that differentiates. We can take human iPS cells and differentiate them specifically into the kidney glomerulopodocytes, which are the specialized epithelial cells in the glomerulus. And one of the reasons why I'm excited about this is the fact that we're able to obtain cells that are developmentally more mature than any other system that we have. So by integrating those into technologies like the Organs-on-Chip platform, we're able to introduce the vasculature that's necessary for establishing that kidney filtration barrier, right, a glomerular filtration barrier, and looking at what happens when you flow a blood or a blood substitute in the system, and whether it's able to selectively filter toxins and other molecules in blood. We don't have that kind of functionality in the traditional organoid system. You can actually filter blood through those systems. So I think we're very early in our ability to design these systems in a way that is beginning to recapitulate some of the early stages of development. But I don't think that it's advanced at a stage where we have functional tissue models that can accurately model the disease, so that we better understand what the disease mechanisms are and develop therapeutics. Because I think if our disease models are far from accurate, I think it's fair to also expect that we might not more accurately develop or identify therapeutics that would be applicable in vivo in a patient, for example. So I think that we do have some ways to go. And again, I just want to emphasize that a lot of these technologies like the organoid is great, but to suggest that it functions like a kidney, it's a bit, it's unrealistic. Yeah.
Right, but let's talk some more about those in vitro technologies, right? Even though we might not be there with the clinical applications necessarily for the iPS technologies and the organoid technologies, your lab has really focused on the in vitro applications, right?
Absolutely.
So you published those back-to-back Nature Protocol and Nature Biomedical Engineering papers a couple years ago when you were in Don Ingber's lab in Boston. And like you alluded to, you're focusing on the directed differentiation of iPSCs into mature kidney podocytes and the establishment of the glomerulus-on-a-Chip chip, like you alluded to. And it's been pretty tough to differentiate mature human kidney cells like you're talking about. So I think your approach was really useful to the field. But then, you took it a step further and actually developed this organ chip. And you're talking about the ways that you might be able to use this chip for, toxin filtration and, in vitro modeling and that sort of thing. But what's the next step of this chip? How can you actually make these glomerulus chips even better? Is it the introduction of other chips that you might be able to connect to the kidney chip, or vascularization? So what's next for these organ chips?
Yeah, so that's a really great question. And I actually kind of wanted to just give you some insight on how this project even started. So we were actually challenged by DARPA when I was a postdoc to create in vitro models of about 10 different organs, and the kidney was one of them. And the group had not been able to develop a model of the kidney glomerulus, because that is the primary site for blood filtration in the kidney. So some other labs, including Don Ingber's lab at the time, had established a model of the proximal tubule, where reabsorption and things like that happen in the kidneys. But we know that the primary function function of the kidneys is to filter blood and remove toxins from blood, and we didn't have a model that could do that. That could basically perform this critical function. So when I joined the team, I just thought, well, why couldn't we I'm trying to just understand why we couldn't do this. And the main challenge was that we didn't have access to functional human kidney podocytes. And there are no commercial sources of these cells. As you can imagine, it requires highly invasive procedure. These aren't self-renewing populations of cells from the kidney. So if they're even derived from discarded kidney tissues, they would either be diseased and not be as useful for understanding what's normal versus disease, or you cannot propagate them to get substantial number of them. So the idea was to establish this method so that we can have access to potentially unlimited supply of these critical cell type for creating an in vitro model of the glomerulus. And so for me, I have to say, it's one of the most exciting projects I have worked on, and it really was enabled also by being in an environment that gave me the independence to pursue a direction that wasn't necessarily the way that things were done in that environment. So I remember Don saying, well, this is not a stem cell biology lab. But I joined this lab with a stem cell biology experience. So when I took on this project, I just asked myself, well, what is known about how the kidney develops? What factors are necessary for differentiating these cells into the desired cell type? And that informed my strategy for developing the method. And so when we were able to obtain these cells and then characterize them and notice that they were so specialized in their function, we thought, well, we have the engineering capabilities in a way that is not even accessible to many in the field. Can we integrate these cells into these microfluidic organ-on-chip systems? We have an idea what the structure of the glomerular filtration barrier looks like. We know we need dedicated fluidic channels for urinary compartment in the vasculature. And so those all helped us design a platform that would allow us to integrate these cells into this chip and then model the function of the glomerulus. So now that we have the model established, in my lab, we're really focused mostly on trying to use this platform to understand human kidney, the mechanisms of kidney disease, and some of the processes that are altered during disease progression. I also envisioned that the platform would be useful for toxicity testing, because we know that about 75% of drugs actually fail during clinical trials, partly due to toxicity to the kidneys. And these are usually not always predictable, or they're not predicted before these drug candidates get into human clinical trials. So I am hopeful that we could potentially use some of these platforms to identify some of these drug candidates that might end up being toxic to humans during clinical trial. We also have projects that are aimed at really extending the engineering capabilities. So we have been able to model one functional unit, the structure and function of the glomerular capillary wall. We know that the kidneys are much more complex than that. So you alluded to strategies where you can develop different models of tissues and then put them together to create a more complex organ. So we're at the stage where we're trying to develop the next generation of these organ-on-a-chip models by integrating complete capillary systems. So instead of just looking at that blood filtration interface or that barrier, we're designing them so we have complete capillary-like systems that would pretty much mimic the whole structure of the Bowman's capsule or the glomerular capillaries in that encased structure for blood filtration.
Yeah, wow. I mean, it's amazing to me because the pace of the progression, it didn't seem like so long ago, I guess, that we were doing these kind of organs on chips, and now we're talking about putting multiple chips together. You know what I mean? It's amazing. And I can say I remember that paper, the Nature BME paper, not only because it's a great story, but also that the journal had just come out. And I remember I was looking at every issue in that first year to see if it was a good fit for some other story that I had cooking. Ultimately, it was not. But I remember it. And I also see that in your CV that you have like a Jove stories. So you're like, you're no stranger to the new media and these new formats. You did a virtual talk at ISSCR 2020. We were covering that. I remember that. You're probably a heavy tweeter, but, looking at your CV, very impressive. You got all the grants, you got all the awards, sure. But also there's like press. You got a lot of press. 100 inspiring Black scientists over here. You got the rising stars in BME over there. You've been winning on a very public scale, prominently winning for a decade now. So for the trainees out there, and for my own curiosity as well, how important do you think that kind of media exposure? Of course, the awards and all the plaudits are important to establish your name and your fundability. But how important do you think the kind of like the media exposure and the general PR are to a successful career path?
Wow. I mean, that sounds like an easy question, but it's actually kind of hard to answer to that, because I think that it's, of course, really important. There's no doubt about it. And I'm just thinking, if I'm talking to a trainee, how do I encourage them to engage in activities that might get them that kind of exposure? And I have to admit that I think that I feel that I have people who would talk about me in rooms that I'm not present. And I think that I've been fortunate to have mentors who've been very supportive of me, encouraged me to really, frankly, just go where my ideas take me and not be limited by where my training left me. And so I think that having people like that who are willing to really tell you the truth and give you that honest feedback and guidance, usually if you're responsive to them and you do well, they would make sure that you get the maybe the acknowledgement they believe you deserve, or at least get the exposure you need to advance your career. So I have to admit that I think a lot of these came to me by surprise, not that there aren't things that I personally initiated. But I do think that it was also not too surprising from the sense that I do try to put my best in everything I do. So I don't, it's not that I don't think it's earned, but I think I do have to give credit to people who are supportive, not just intellectually, but also mindful of how that their support impact my career advancement.
So let's talk about those mentors, right? So you did your postdoctoral training in snowy Boston, in George Church's lab, and also in Don Ingber's lab, as we were alluding to. And Don Ingber in particular is really a pioneer in bioengineering, even though he might not necessarily be a stem cell biologist. He's a bioengineer guru and icon. And it seems like so many of the top organ-on-chip papers have come from his lab. And a lot of these engineering developments from the Ingber lab have turned into products and companies and in hopes of actually improving the field of in vitro toxicology and maybe ultimately totally replacing the mouse as a model system. That would be kind of a dream, right? So tell us more about that time in Boston and your time as a postdoctoral fellow at the Weiss Institute. Mm-hmm. So what was it like to actually train in the Ingber lab?
As a scientist, I think being at the Weiss it's like the ultimate playground. For me, I think I was blown away by just being in an environment where you can be challenged to do something, but then also supported in every way possible. If you come up with too many bad ideas, that wouldn't work out too well. But But Don was very supportive, and I remember even when he wasn't sure what direction I wanted to take something, if I communicated to him how I'm thinking through it, and then just laid out a rationale for why it's worth giving it a shot, he would just be like, okay, go try it. And I also have this habit of setting timelines for myself, saying, okay, I'm going to give this a shot for maybe about 4, 5, 6 months. If it doesn't work, then we're going to have to maybe refocus. And I think a combination of that and having the resources to pursue some of these directions was just incredible. And I think Don is also the kind of person, and both him and George Church, I mean, one of the things they always advised me on is to just kind of not being afraid of failure, right? So if you are going to try an idea, yeah, there's a chance it might work, but you should be comfortable with the fact that it might fail, and then just think about how you use that information to guide the direction you ultimately end up taking the project. So I think just being in an environment where failing is like not it's not necessarily a bad thing, because some of the most important projects and some of the most exciting directions that they've taken have usually come out of projects that initially maybe failed or perhaps didn't work out the way that they were expected to. And so to me, that was a really powerful realization, and being in an environment that's supportive of that was absolutely critical. Yeah, and I can't stress enough how they are support. I think if you do ask, they would be there, and they would help. They provide you with the feedback you need. They're very direct, and I think to me that was very important. Just having mentors who can communicate directly. I didn't have to spend too much energy trying to guess what they're thinking, and so I think it does help with being productive and having a sense of direction when you're not so sure where to go or which path to take.
Yeah, I can imagine. Arun had a similar experience over there in Cambridge. I can imagine it must have been very exciting to make that move. This is no shade on Wisconsin. I know, it's the birthplace of human ESC research, right? But Cambridge, right? That's where dreams collide with reality, and that's for better and worse, as you just alluded to all those failures. But, on the path towards, resounding success most of the time. And then there's you with this big paper and the patent I saw you got off of that. Maybe some talk about licensing or like starting a company. I know a lot of people they have a big paper out of the beast, and suddenly there's a company. So as someone who's still kind of at the beginning of their career, you could have bent more towards an industry biotech path if you wanted to. You could have been the CEO of something, your own company. Do you have any insight to share again to the people behind you maybe who might be in a similar position? Are there any pitfalls you think there's kind of a trap to, starting a company as opposed to staying on the academic path? Or maybe do you aspire towards those kind of commercial ends alongside your academic ambition?
Yeah, absolutely. Alongside the academic ambition, for sure. But I think at heart, I feel like I've always been an academic, and I can't really imagine my life without being in science and academia, or having some of the flexibility that I think academia affords scientists to be able to explore some of these questions that excite you. Of course, you have to be able to get funding for it, but I think it affords us with a level of independence that I personally think I find attractive, at least attractive for me intellectually. Most of my interest in industry is really being able to see technologies come out of the lab and not just stay in the publications and on the walls, but really hopefully change patients' lives in the future. So you notice that at least for me, for every project in my lab, And for every new person who joins the lab and we're thinking about what project they work on, I always ask, who is going to benefit from this? Who could potentially benefit from this science? And how? And how would it change the field? So some of these are usually lead-in questions and how we, what projects I even prioritize or decide to focus on, and what kind of questions we ask when we're pursuing the science. So yes, I do have interest in the commercial or the technology transfer or translation aspect of science, but I think I would always be a scientist first.
So even though you're an academic first, you're at an amazing place to kind of bridge that gap, right? To bridge the gap between basic science and translational biology, right? Yeah. You're at Duke University, and it's no secret that we're fans of Duke University on the show, which is, of course, where Daylon and I did our undergrad studies back in the day. And we've actually had a lot of PIs on the podcast who actually have connections to Duke in some fashion. We had Joy Wu, who I believe she did her part of her medical training at Duke, and Todd McDevitt, who I think was an undergrad there as well. So we've had PIs who have spent some time in Durham at some point in their training, and you decided to set up your own lab at Duke. And it seems over the last few years, Duke has actually really boosted its research prospects in stem cell biology and regenerative medicine, especially with the development of this new Regeneration Next initiative. I think that's right.
Yes.
And I think that's led by Ken Poss. Yeah. So Daylon and I could go on forever about our time at Duke and about the amazing food in Durham, but as a new PI down there in the Research Triangle, what have you loved about your time at Duke University, and what makes it a great place for stem cell biology and bioengineering as well?
Absolutely the people. The people are amazing, and I think that just being able to collaboration-wise, I think we go from talking to actually collaborating on projects fairly quickly. And the proximity of the schools, medicine and engineering, are quite literally about 4 minutes walk apart. The basic science, the biological departments and cell biology, they're all really centralized in terms of location. So I think it really makes it fun, whether you're a basic scientist or thinking about what a clinician thinks about your science, or how they would whether they would be interested in using a technology you're developing. They would be right down the hallway for me to talk to them. And so I think it really, for me, it really does make it an exciting environment to be to do science. And other things I've really loved about Duke, my initial impressions when I visited Duke was definitely how beautiful the campus looked. And I remember just driving and thinking, I would really love to come to work every day, just even if it's just to see this view. And so it's quite pleasant. I think just kind of loving the environment itself. I find that it boosts my energy. It definitely makes it really exciting to come to work, and my colleagues are absolutely amazing. And for me, being in engineering and medicine has been absolutely critical, really, for some of the directions that I envision taking in our lab, from both the basic science and engineering perspective, to hopefully get more translational with our work. I think being affiliated with both departments and schools really provides us with the resources and the type of interactions with colleagues that we would need to advance some of these technologies. But I also find that nephrology, for example, here is very excited about the technologies that we bring to the field. And so I think that also contributes to how we collaborate with folks, and people have some very challenge and problems they've been wanting to address, and maybe they want to look at it's relevant to humans and actually using human tissues. And so there seems to be natural synergy in what we do with what others are also hoping to accomplish. So I think all of these together really, truly make it an exciting environment. I can go on and on about what I love about Duke and Durham. Honestly, I love the greenery. I mean, I lived, so I went to graduate school in Madison, Wisconsin, with Laura. Laura's now at MIT, actually, but I started with her when she was in Madison, Wisconsin. It was, of course, very cold. I thought Binghamton had prepared me well to live in Madison, but not really. I realized that most of my winter jackets were turned into spring jackets when I went to Wisconsin. So when I moved to Boston, Boston was actually relatively warm for me, and but everybody complained about how cold it was in Boston. And so over time, of course, I also adapted and started complaining that Boston was cold. So that all made North Carolina even more attractive. and then I really love greenery. I love it. I think we live in the woods pretty much, and just part of my joy for the day is just driving through the woods to and from work.
Yeah, Duke, so beautiful as I remember it. Yeah, we could go on and on, although now that my sons are approaching the age where they could get themselves into trouble, my memories would have to be, or the record at least, heavily redacted. Okay, we don't want to go too deep there. I'm exposed a little bit. But I also remember very vividly about my time at Duke that people would totally fall. You know, I was pre-med by way, to get into grad school for science. Most people were going to med school, but all my pre-med friends who then went BME, they just disappeared socially. They just went away. That's how hardcore the BME major was. But now I can see that they had the last laugh because they got to work with the likes of you. So ha for them. But along those lines, Arun mentioned Regeneration Next, and a major pillar of that effort is the promotion of an inclusive and diverse space in the research triangle. And you're, originally from Ghana. You've been in New York. You went to Wisconsin, Boston. You've been all over the place, so you can take care of yourself. But what's your expectations, or do you have any kind of apprehension about the role, the outsized role that race plays in that atmosphere?
Yeah, I mean, that's as a Black scientist, I think I definitely am very aware of these challenges. It's been nearly everywhere I've been, even in cities that might actually be more diverse. The institution itself, or maybe I might be the only one in my department, or sometimes in the lab, or so I've been in a lot of places where just sometimes inside or in the department might not reflect the population outside of the school. And one of the things that I've realized over time is that you might have to do more as a Black scientist to kind of prove yourself. But once you establish that, people could start recognizing the science. And I think at Duke, I'm of course fairly new to Duke, but I feel that Duke I've been impressed with some of the initiatives that they have and some of the resources that they're providing to not just faculty, but students and staff. So I think overall it's a very, very supportive environment. And so in so many ways, even though it might be historically in a location that has been viewed a certain way, I think they've made some very progressive effort and implemented some initiatives that I hope would make it a much more welcoming environment for everyone. I can't speak to what I do in my lab because I think at the university level, I don't really have a lot of control over that one. But in my lab, I really do make sure that I encourage open communication and make students very aware, or try to make them aware of some of the experiences that everyone brings into that space. Like some students you may see a scientist who is an African American, but even if they've been very, very successful, they might have had to deal with or experience or go through some very challenging circumstances to get to where they are. So kind of being sensitive and informed, or kind of being open to being educated about some of these challenges that individuals face because of differences in background, I think is very important. And I encourage that in my lab. And I think that doing that has helped us as a group to be more open and also kind of identifying when things happen and it doesn't align with our values in the lab. And I think many times people really know what the right thing to do is, but they just have to either care enough to act or know and have some strategies on how to approach things. And so I find all of that important. I should also point out that when I was thinking of where to go to for faculty position, I found that even though I might be the only one of very few in my department, that across Duke, there are highly successful Black scientists all across the campus in different departments. And so for me, that was really telling, or at least it made me feel that, I wouldn't be if I come here and I'm successful, I wouldn't have to just think about being the only successful African American. So it mattered to me that I see other African Americans, whether they were in my department or not, that they've been successful here at Duke, and that was true in multiple departments. Fortunately, I actually did notice that when I was interviewing, and that also did influence my decision to come to Duke, actually.
Yeah, I think visibility is really everything.
Yeah.
And as a recent guest on the podcast, Natasha Muwanigwa actually said, and just seeing that you are a successful African American woman scientist is an exceptionally powerful thing. And as a recent alum of Duke University, I can echo what you're saying. For me, at least, it was nothing but a very diverse and very welcoming environment for students and also for faculty. So I think it's been an incredible place to be. So thank you so much for joining us here today, Samira. It's been a pleasure talking to you, and not only just because you're a scientist at Duke University, but you're also an exceptionally accomplished stem cell biologist and working in a very hot topic right now in tissue and organ engineering and organ-on-chip biology. So before we actually let you go, we're going to ask you a couple of science peripheral questions. So starting off, what non-science book are you reading or that you've read recently that's awesome and you want to share with our listeners?
Okay. So one that I really, really love, and I have reread this. I try to reread this whenever I'm making a big move in my career, has been The Four Agreements by Don Miguel Ruiz. And it's fairly simple. It's just it's four agreements, but it's actually I think is really, really powerful. And I can maybe just briefly just tell. So one being impeccable with your words. The second, not taking anything personally. The third being not making assumptions. I find this very important for science as well. And always doing your best. So for me, I think these are really powerful kind of guiding principles, both at the personal level, but also professionally. I think that almost every time something happens or goes wrong or seems to be going wrong, it appears that one of these things have been violated. So one of these rules have been violated. So I think that it's truly a powerful book and just concept to have and think about regularly as a person, both at the individual level, but also professionally, and how we interact with people as well.
Well, it's clear, at least to me, that you're really an adherent to the first of those principles. You are impeccable with your words and speech. It's fun to listen to you. So we'll ring one more series of answers out of you. Very quickly, fill in the blanks. First, the biggest thing in the stem cell field right now is?
I think the ability to model human biology and understand how humans respond to drugs. I think it's really the direction that stem cell biology is going to make a huge impact in.
First clinical inkling's probably going to be there. I agree. Next, I would never have gotten to this point in my career without?
My mom.
Oh, that's very sweet.
Absolutely.
Well, she deserves the credit.
Absolutely. And I can quickly just say that nobody could have instilled the value of education in me better than she did. So I absolutely would not be here without her.
Mom, ditto. I said that to you too, okay? Next, when it comes to blank, I am pretty much useless.
Yard work.
Yeah, well,
never.
The yard will be fine.
I love when it looks nice, but no.
Well, you have a young child, I hear. That's what you made that one for. Last, if the lab catches fire and I have a chance to grab one thing on my way out, it's my
students.
Oh, there you're just a real sentimentalist.
This is something that I actually do. I mean, I know that they can move, but I actually, whenever we have the fire, if we have a fire drill, or even if it's just a test, I always walk. I cannot leave the building without checking whether they've left. I go in the lab. If there's anybody in there, they have to leave with me.
That's so sweet. In fact, I don't think anybody's actually given that answer, which I don't know if that says something about our guests. I don't know.
Well, we're all differently abled. Maybe, some cases they're more invested in their trainees' ability to get out of the lab themselves. But it seems like Samira doesn't have that faith.
Yeah, I'm curious what the most popular answers were. I guess lab notebooks could be important, but but I think
you get a lot of laptop. It's all from the boomers who aren't aware of the cloud.
Well, these days, if you save everything on the cloud, you'll be fine without your laptop.
You don't know. There's hackers out there, Samira. Watch out. Anyway, thank you so much. This has been a lot of fun for us. We hope it's been fun for you and for the listeners for sure. You'll have to come back soon to give us an update on your really meteoric rise in the field. We'll be excited to hear that.
I would love to. Thank you so much for having me. This has been really fun. And I truly do appreciate also that this is the Blue Devils edition. So it's great meeting you both.
Yeah, it won't be the last.
Yeah. Yes.
All right, you guys, that brings us to the end of this episode. Don't forget to subscribe to our newsletter at www.stemcellpodcast.com to get the show notes, including an episode summary and links to all of the interview and Roundup papers. You can also reach out to us on Twitter at STEMCELL or via email at info@stemcellpodcast.com with feedback or to suggest guests. Thanks again to Samira for joining us today on the show. Remember, guys, we are taking a short break, but we will be back on Tuesday, January 12th, so tune in then. Thank you for listening.